Okay, so this is my presentation on organic chemistry. We'll be looking into organic molecules, in particular, organic molecules. To understand so our learning objectives are to understand the IUPAC, nomenclature of organic chemistry, to understand and identify organic structure, and to understand and identify the important concepts of organic chemistry, including functional groups and the homologous series. Now, this first slide is actually the naming and formula of organic compounds. We have the compound category, which are the ones we're going to be looking at today, the functional groups they contain, the structure of the functional groups, and the suffix. Now, this is basically the name ending for each compound. The second table shows the number of carbon atoms that are present in a compound. Now, this will basically go on how the name is. So if it has one carbon atom, for example, it will begin with meth. If there is a side chain present, it will actually begin methyl. And the multiplier just indicates how many of these side chains are there. So if there were 2, for example, it would be diethyl, and then whatever the ending would be for that particular compound. The naming and formula, there's more into it as well. So we have the homologous series. If it's part of that group, it just basically means that they have similar chemical properties, but they differ in the number of CH2 groups. There's different ways of showing this. We've got the molecular formulae, empirical formulae, displayed formulae, and structural formulae. We also have skeletal formulae. Now, the point to remember with skeletal is that it looks just like lines. However, at each point of where the line is, that actually consists of the carbon atoms. The displayed formula and the structural formula, they're very identical in its presentation. However, the structural formula can also be condensed into a written version, which you'll see here as CH3, CH2, CH3. Structural isomers. Now, these have actually got 3 primary ways in which a structural isomer can occur. We have chain isomerism, positional isomerism, and functional group isomerism. So chain isomers occur in compounds with the same molecular formula, but the atoms of the hydrocarbon chain have different structural arrangements. This results in a branched or unbranched, which you can see from the example. Positional isomerism occurs when the atoms of the functional group within the compounds with the same molecular formula are displayed in different positions along the main hydrocarbon chain. Functional group isomerism occurs when the compounds have the same molecular structure, but the functional groups are different, as seen in the example below as well. We then have further ones. So stereoisomerism refers to compounds with the same molecular and structural formula, but the atoms occupy different spatial arrangements. Now, there's 2 types of stereoisomers. We have geometrical and optical. Geometrical, so they can only occur in molecules which contain a carbon-carbon double bond with 2 different groups attached to each carbon. Now, these can basically be defined as cis-trans or EZ isomerism. The naming schemes of them, these are the naming schemes. So cis-trans isomers contain 1 type of functional group, whereas EZ isomers contain more than 1 type of functional group. Optical isomerism can only occur in molecules of a compound with a chiral centre. Now, a chiral centre basically means that the carbon atom is attached to 4 different atoms or groups of atoms. And for each chiral bond, only 2 optical isomers are formed. This structural arrangement results in optical isomers. They're actually non-superimposable mirror images of one another. They are basically referred to as enantiomers. Optical isomers, so they're chemically identical. However, one rotates polarised like clockwise and the other one anti-clockwise. An equal mixture of these 2 enantiomers are known as racemic mixture. The naming of them actually depends on the arrangement of the molecules, which you can see in the example. Now, to distinguish the correct names between enantiomers, you go by something that they call the Corny rule, and that just basically means you can spell corn, and it depends if you go around clockwise or anti-clockwise. So the D amino acid is the anti-clockwise, and the L amino acid is the clockwise. Now, alkanes are hydrocarbons. Now, this means they're basically only formed from carbon-hydrogen atoms. They're a member of the homologous series. They contain the alcohol functional group. They're classified as saturated compounds because they only have single covalent bonds between carbons or carbon and hydrogen. And alkanes with four or more atoms display structural chain isomerism. As you can see in the examples below, it can result in either branched or chained molecules. Alkane names, they always end in -ane, but in terms of the beginning of their name, it depends on the amount of carbon atoms, which will determine their name and if there is a side chain present. And you can see here there are quite a few physical and chemical properties. Alkenes, now they're also hydrocarbons. They're another member of the homologous series. They contain the alkenyl functional group. Now these are formed from carbon-carbon double bonds, so they're classified as unsaturated compounds. The double bond of alkenes is actually comprised of two parts. We have a sigma and a pi bond. Now the pi bond is generally weaker, so this one's relatively easy to be broken, allowing other atoms to be attached to it. The naming is very, very similar in terms of counting the number of carbon atoms, and that will determine the beginning of the name. Alkene names will always end in -ene rather than -ane. So there's quite a few reactions, so they can undergo addition reactions, and there are a few listed below with an example just here of the unsymmetrical alkenes. And there are physical and chemical properties as well, which are listed to the right. Alcohols, now they are a member of the homologous series. They contain the hydroxyl functional group. Their classification of alcohol depends on the number of alcohol groups bonded to the carbon carrying the OH group. Now, alcohols can actually be specified into three different categories. We have primary alcohols, secondary alcohols, and tertiary alcohols. So you can see from here, primary alcohols can be identified by the attachment of the carbon atom to the OH group, and either no alcohol group or only one alcohol group will be attached to it. They can oxidise in two steps, and the first is they can oxidise to an aldehyde, which then gets oxidised to a carboxylic acid. Secondary alcohols can be identified by the attachment of the carbon atom to the OH group and two alcohol groups. Now, they can be oxidised to ketones in a similar oxidisation process. However, the ketones cannot be oxidised any further. Tertiary alcohols, they can be identified by the attachment of the carbon atom to the OH group and three alcohol groups. Now, tertiary alcohols are not readily oxidised as there's no hydrogen atom that is bonded to the carbon, so no oxidisation reaction can take place. Now, aldehydes is our next one. These are carbonyl compounds, which can be defined by a general formula, CnH2NO. Aldehydes contain the carbonyl functional group, which are formed from CHO bonds. These contain a carbon-oxygen double bond, and they can undergo various reactions such as reduction, oxidation, and nucleophilic addition, which we have some examples of. The naming of aldehydes is very identical to the naming of alkanes, except the suffix ends in -al. Aldehydes contain at least one hydrogen atom, which is joined to the carbon atom of a carbonyl group, displayed at the end of the chain. So when determining the prefix, we count the carbon atoms, and that will determine the beginning. And we also, so because of the carbonyl, when we determine, the prefix, the counting of carbon atoms starts from the carbonyl group in the unbranched chain. So the presence of a side chain should also be taken into consideration, because you can have unbranched or a branched aldehyde. And again, we have some of the chemical properties to the right. Ketones are, again, carbonyl compounds. They have the carbonyl functional group. They can undergo various reactions again, such as reduction, oxidation, and nucleophilic addition. The naming of ketones is also identical to the naming of alkenes again, but instead these ones end in -one. So they contain two other carbon atoms or hydrocarbon groups, which are attached to the carbon atom of the carbonyl group. We have a couple of examples at the bottom, and we have the formation and reaction of ketones. So oxidation of these refers to the process that forms ketones by use of an oxidizing agent. So secondary alcohols, like we looked at before, oxidize to form ketones. However, like I said before, ketones cannot be further oxidized. Reduction of ketones refers to the reverse process. So these ketones can actually be reduced back into secondary alcohols by use of a reducing agent. And we have some of the physical and chemical properties to the right again. Carboxylic acids and esters is the next one we're looking into. So carboxylic acids are another member of the homologous series, and they contain the carbonyl functional group. They can undergo reduction reactions to form alcohols via a powerful reducing reagent, and can also undergo reversible reactions to form esters. Hence why I have got these on one slide. So the naming of carboxylic acids is identical to alkanes again, but it ends in oic acid. Carboxylic acid formation and reactions. I have a couple of examples there, and they can actually react in the same way as usual acid reactions, and these can actually form carboxylate salts, which I have some examples of. Esters are formed from the compounds of the homologous series, so they're actually formed from carboxylic acids and alcohols in a process known as esterification, and they contain the ester functional group. The naming of esters follows the way the esters are formed, so from the alcohols and the carboxylic acids via an acid catalyst. So the alcohol determines the first part of the name, and the carboxylic acid determines the second part, which you can see. And esters always end with the suffix -ate. Amines, they are compounds containing nitrogen, which are obtained from ammonia. They can be classified as either primary, secondary, tertiary, or even quaternary, depending on how many hydrogen atoms in the ammonia have been replaced by another organic group. So we have primary amines; they have been replaced by one of the hydrogen atoms have been replaced by another group. Secondary amines means two of the hydrogen atoms attached to the nitrogen is replaced by another organic group. And tertiary amines can be identified by the structure as three of the hydrogen atoms attached to the nitrogen being replaced by another organic group. Amides. So amides are organic compounds containing nitrogen again. They are obtained from carboxylic acids. They contain the carboxylamide functional group. So this is formed of CONH2 bonds, as the NH2 group replaces the OH group of the acid. So the naming of the amides depends on the carboxylic acid that's used in the formation reaction. So here we have an example of ethanoic acid plus ammonia, and that will become ethanamide. The formation and reaction of amides. So we have acid hydrolysis, base hydrolysis, and dehydration, with some examples there for you and what they form. Physical and chemical properties again can be seen to the right. So identifying unknown compounds, there's various ways we can actually do this, but one example that we're going to look at is the qualitative analysis of alcohols, aldehydes and ketones. Now, this is done in an experiment which I have done a sort of example of how to do it. So we've got what reagents we would use in order to determine what compound is present if it's in an unknown compound. So if you look to the right, you can see a diagram tells us that we have an unknown compound, and we then put in certain reagents, and certain compounds will react to these reagents to determine the names and what is present. We also have a table of results to show what we found. There is just a little description of what each reagent is and how each compound reacts to that. And we also have some images just here to show you. Now, the importance of simple organic molecules when studying biological molecules is our next slide. So there's three most important organic molecules that occur naturally in living organisms. Now, life can't actually occur without these. The main three are carbohydrates, proteins, and lipids. They each have their own roles within an organism to ensure survival and correct internal functioning. Each of these organic molecules consist of different types with different functions. So we have carbohydrates. Now, these are classified as an important molecule because it provides energy and structure to most of the body's cells and tissues. We have some examples. So we have monosaccharides, which are simple sugars such as glucose and fructose. And then polysaccharides, which are long chains of these monosaccharides, resulting in starch, glycogen, and cellulose. Proteins are classified as another major organic biological molecule. So proteins are actually really important because they build up the structure of a living organism. They're mainly made from amino acids. They are the building blocks, and their main roles are things like acting as antibodies to fight disease, control and maintain hormones. They transport oxygen into the blood. And then a really interesting one is that they can actually be further defined into primary, secondary and tertiary structures, all of which actually play significant roles in DNA, which determines genetic information. Lipids they're classified as essential because they have a variety of roles within a living organism. The most common lipids are fats and oils, steroids and phospholipids. Fats and oils protect organs. We have animal and vegetable oil are esters called fatty acids or triglycerides. We have an example for each of these compounds just at the bottom here. So we've got glucose, fructose, amino acids, and a structure of glycerol, which is a lipid. And that's everything. So this is my reference list. Thank you for watching. Done.